Hello students, welcome to this third video in the series on cell structure and function. And in this video we'll be covering the endosymbiotic theory. And we'll be first looking at the structure and anatomy of these three cellular organelles, which is the mitochondria, chloroplast, as well as peroxisome. And then we'll be culminating in the endosymbiontic theory. So without further ado, let us begin. So first we need to know what are the mitochondria and what are the chloroplasts. So let us start with the mitochondria. Mitochondria are the sites of cellular respiration. And in cellular respiration, what happens? Sugars are broken down and ATP, adenosine triphosphate, which is a form of energy, is released. And this process is called cellular respiration. And the site of this process happens in the mitochondria. And this is very important. Why? Because the cells that require energy, which all cells do, and the cells that especially require to contract, for example your muscle cells, they are very rich in the mitochondria because the mitochondria is the source of energy for all cells. And how does it do this? By breaking down sugars. So let us look now at the structure of the mitochondria. So first important thing is that there are two membranes. First is the outer membrane, and second we have the inner membrane. So this space in orange here is the space between the outer membrane. The outer membrane is actually very smooth and the inner membrane is very convoluted. It has a high surface area to volume ratio. So the space between the outer membrane and the inner membrane is called the intermembrane space. And the space inside the inner membrane is called the matrix or the mitochondrial matrix because it is inside the mitochondria. So what all is inside the matrix? Firstly, we can see here that ribosomes are inside the matrix. And actually what makes the mitochondrion special is that it is one of the organs that also contains DNA inside the cell. We call this mitochondrial DNA. And this DNA is actually circular DNA. And this circular DNA is called a plasmid. Usually we find plasmids in bacteria, but it is also found in the mitochondrion. So now we need to remember that there are ribosomes and DNA in the mitochondrial matrix. So what can ribosomes form from DNA and mRNA? It can actually form proteins. So inside the mitochondrial matrix there are a lot of enzymes that enable respiration. And as well as embedded in this membrane, the inner membrane, there are a lot of proteins that engage in respiration. So respiratory proteins. So because this inner membrane is very convoluted, there is a lot of space for the respiratory proteins to be embedded in the membrane, which means you can have a lot of respiratory proteins here. And this means that the efficiency of respiration goes up because of this convoluted nature. And we call this high surface area to volume ratio. So now let us move on to the chloroplast. So why is the chloroplast important? You may have heard of the process of photosynthesis. Chloroplasts are enabling the photosynthesis in the plant, and the importance of photosynthesis is because it produces sugars in the plant and it uses solar energy to do so, and the raw material is actually the carbon dioxide and water in our air. So this is quite a magical process, and therefore the structure of the chloroplast must be specialized to do that. So the first thing we notice also that the mitochondrion has two membranes. The chloroplast actually has three membranes. So the first membrane is the outer membrane. The inner membrane Now, unlike the mitochondrion, both the outer and inner membranes are all smooth. So there is actually a third membrane. You can see all these stacks inside the chloroplast, and this is called a thylakoid membrane. So it actually looks like many coins stacked together, but all of this is enclosed by one membrane that is continuous throughout, which means all these stacks of coins are connected. So each coin in the stack is called a thylakoid. And the stack itself, this whole stack, is called a granum. In plural, it is called grana. So that is the structure. So where is the chlorophyll inside the inside the chloroplast? It is actually inside the thylakoid membrane, inside the granum. So inside the thylakoid membrane, there's a lot of enzymes that enable photosynthesis. So that will be all here. And in addition to that, there are enzymes embedded in the membrane of the thylakoid membrane. The chlorophyll, the pigment that enables photosynthesis, is also inside the thylakoid membrane. And it is this very substance that enables photosynthesis and causes the thylakoid membrane to become green. So the thylakoid and the grana are the sites of photosynthesis. So now there are other regions in the chloroplast as well, which is this intermembrane space. And this is called the stroma, this yellow part. Much like the mitochondrion, inside the stroma there we can also see that there are ribosomes. And there is also circular DNA. So the mitochondrion and the chloroplast are quite similar in this aspect. So I'll write here ribosomes and circular DNA. So all the enzymes that enable photosynthesis will actually be in the stroma because the ribosomes are producing the enzymes here. Okay, now we will move on to the peroxisome. So you may not have heard of the peroxisome, but it is actually a very important structure, especially for those alcoholics. Because the alcoholics who taken a lot of alcohol is actually toxic. This is ethanol. You need to detoxify it. So it will actually become this. And this hydrogen will go onto an oxygen atom and it will become H2O2. So this H2O2 is also toxic. So it is inside this peroxisome where the H2O2 is converted to H2O. So we can see it in this reaction. The hydrogens from toxic substances go onto an oxygen atom and it becomes H2O2. And the H2O2 by some enzymatic reaction is also converted to H2O. So now that because of that the toxins become non-toxic substances. So you can see here that this is the peroxisome. It has one single membrane, whereas the mitochondria and chloroplast have two membranes and three membranes. This is only one membrane. And inside you can see this red color core. So this is actually a crystal core. This crystal core is actually a large crystal of enzymes that catalyze this non-toxic reaction. Scientists are still finding out more things about the peroxisome, but we know that it is usually clustered together with the chloroplasts and the mitochondrion. This is because it catalyzes fatty acids conversion into sugars. So these sugars can travel from the peroxisome to the mitochondrion, and the sugars now can be used in the mitochondrion for respiration. Another function of the peroxisome is to take all the toxic metabolic products from the chloroplasts and the mitochondrion and convert them into non-toxic products. So mainly it functions in detoxification, and that means that we can find this in high concentration in the liver. because the main function of the liver is in detoxification. So now we have looked at the structures of the mitochondria, chloroplasts and the peroxisome. So now let us look at this endosymbiont theory. So this theory covers by scientists who questioned how come there are these mitochondria, chloroplasts and peroxisomes coming into eukaryotic cells. So the scientists look at a few factors and the characteristics of these three organelles. Mainly they looked at the chloroplast and the mitochondria. So the first thing is that both of these organelles have double membrane. Second thing is that inside this they had ribosomes as well as some DNA. And this DNA was in the form of circular DNA, which is called a plasmid. And the third important thing is that a cell divides by itself, but these organelles are considered semi-autonomous. This is very important. Semi-autonomous. And this applies to all three organs here. This and this. And because it is semi-autonomous, we say that it can divide by itself. So at random times inside the cell's life cycle, the chloroplast will pinch itself into half and it will become two different chloroplasts. And the same will happen for mitochondria and sometimes for peroxisome. So the scientists figured that these actually have a lot of the characteristics of the really old bacteria. Old bacteria or prokaryotes. So they figured that if there was an ancient eukaryotic cell, old eukaryote, for example maybe one amoeba, a really old one, maybe it could have engulfed this by phagocytosis. This is a mitochondria and this is a chloroplast. And last time they figured that this could be one of the old prokaryotic cells that function by itself. So for example, this could be a photosynthetic prokaryotic cell, and this could be a prokaryotic cell that was very efficient at converting sugars into energy. So if this old eukaryotic cell had engulfed these two, and maybe it had failed to digest it, then these bacteria would have become the endosymbiont. Endosymbiont means a cell living within a cell. And eventually, after many generations, this would function in conjunction with this cell. So for example, now the mitochondrion, which is present in all animals, all plants, all life actually, that is not prokaryotic. Most eukaryotic cells have the mitochondrion. The mitochondrion actually serves to provide energy for the cell. That's why it is called the powerhouse of the cell. And the chloroplast could have been a green, the chloroplast was actually first found in green algae. And this green algae, the ancestral green algae, could have possibly engulfed a photosynthetic bacteria. And it could have become this endosymbiont. So this could have led to the presence of mitochondria and chloroplasts in our cell today. Thank you. And I hope you have enjoyed this video. If you have enjoyed this video, please press the like button and click subscribe for more such videos. Thank you.